Hello everyone and welcome to the Special Senses lecture. First, we need to start by defining what a special sense is versus general senses. General senses are going to be things like touch, your ability to perceive temperature, and your ability to perceive pain. Sensory receptors, such as the various little nerves that you're going to find underneath your skin, are going to allow for your body to respond to stimuli from your external environment and then convert that into nerve impulses. So your ability to know that your fingers are touching something, that they're being pushed against, or maybe, something painful, or, temperature. That's going to have to do with those little nerve endings at the surface of your skin, as well as those corpuscles that we discussed when we were talking about the skin and skin accessories. I do want you to know these two terms, sensation, which is how the brain interprets the impulses. An example would be like interpreting temperature, so hot or cold. And adaptation is another term I want you to know. This is the process by which the magnitude of the receptor potential decreases over time in response to a continuous stimulus. A good example of this is wearing clothing. You kind of go a little bit blind to wearing your clothing once you've worn it for a long time. So you probably perceive wearing your clothing as soon as you put it on, but through the day you kind of forget in a way that you're wearing clothes. The same thing can be said for going like nose blind to something, if you want to think of those Febreze commercials. Young man's room might smell quite bad, like BO and other nasty things, but he can't really perceive it because he's been living in that environment for so long, he doesn't perceive it anymore. But of course, someone who hasn't lived in that environment, they'd be hit with that wall of smell. So those are the general senses. We want to talk about the special senses. The special senses are going to be smell and taste. Those are your chemical special senses, hearing, equilibrium, and vision. One of the things that kind of gets misinterpreted when you learn about the senses when you're younger is that it's smell, taste, hearing, vision, and touch. Touch is a general sense. It is not a special sense. Tactile sensations are not special senses, so make sure you know that. But what goes along with tactile perception, if you will, is itching and tickling, which are kind of interesting things that go with that. An itch is going to be several different tactile sensations kind of mixed into one. It's a combo. It is a sensation that makes you want to scratch. Generally, chemical irritation can cause an itch, but it can also be induced. So as we're talking about itching or itchy or something that's scratchy, making you want to itch, somewhere in the back of your mind you might start scratching, thinking about scratching, or if you see someone scratching, you might start doing it. Maybe you're unconsciously kind of doing it right now. And then tickling is another one of those sensations that is going to be kind of a combination of sometimes pleasure, pain, as well as irritation. I find it quite unpleasant, as most people do. Tickling is not that fun. But it's actually not very well understood as to kind of like the mechanism of tickling and how that is sort of perceived and how it goes about being. Like, you're more ticklish maybe around your neck, maybe under your armpits, your belly, your feet. Various places on various people can end up being very ticklish zones, but not on others. So it's not that well understood, but kind of an interesting thing to bring up. So here we're going to start with your sense of smell. You're going to have something called olfactory receptors, which are going to be known as chemoreceptors, meaning they are chemical receptors. They will receive odorants, chemical smells, and be able to take that up to the olfactory bulb. This is going to be above the olfactory organ, but you don't really need to know that. Just tuck it away for later. information is going to be processed here in the olfactory bulb before it is sent to the cerebral cortex. The olfactory bulb is going to be this little projection that is a projection from the olfactory nerves, which is going to be the nerves that are on the anterior part of your brain. And this olfactory bulb is going to have these little nerves coming down through the cribriform plate of the ethmoid bone. And what's really quite interesting, if you remember this cribriform plate and how it looked when we were looking at the skull, is it was porous, and that is because it's allowing for these nerves to come down and go into your nasal cavity here. So kind of cool. So let's talk about these smells that you're able to perceive. Putrid, like rotting fruit; floral, flowers, right? Peppermint or mint; and musky. And I didn't have a great picture for it other than kind of this natural smell that people can get, and that's not necessarily, I'm saying body odor. This is sort of this natural smell of like sweat, so a muskier smell. There's actually quite a few perfumes and colognes that are going to have a muskier note to it. A lot of odors that you perceive are combinations of these four primary scents, which is kind of interesting to think that everything that you've ever smelled really is just a combination of all of this. A few things to kind of make note of is that humans are far less sensitive than many other animals to scents, and some individual humans can distinguish several thousand different odors, while most humans can easily identify just several hundred. So some people might be more sensitive to smell than others. I've always been a little bit more on the sensitive side, but I also have a very sensitive sense of taste. So it kind of makes sense that you'd be sensitive to smell as well, because they kind of go hand in hand. A few more facts about your nose and kind of just in general the olfactory region is that most olfactory information is going to pass through the limbic system. This is a part of your brain that is strongly associated with emotion and memory. In fact, it's been claimed that olfactory memories are stronger than most any other type of memory, so audio or taste or visual. So certain smells can really trigger some long-term memories, and it's actually been shown that like your hometown type of smell, or maybe like a smell from when you were a child, can really just like snap you back into a memory that you didn't know you remembered. Kind of cool. Just like our sense of taste, our sense of smell is instinctual. So sweet smells and cooking meat is going to trigger pleasure receptors in your brain. And rotten smells like sewage and spoiled food is going to trigger a response of disgust. The term ansomnia is a complete lack of smell, and something that I like bringing up is the term pheromones. These are nonverbal signaling among individuals of the same species. In humans, the ability to sense pheromones is very, very limited. And I always think of this woman trying to sell me a candle, and it was supposed to be a more intimate candle, for intimate moments and things like that, kind of to set you in the mood, if you will. And the thing that had to do with this candle is that it was infused with the human sex pheromone. There's no such thing. You cannot smell someone's sex pheromone. You can't tell if they're ready for sex or anything like that. That doesn't exist in humans. In other animals, there is something kind of like that, something kind of similar, but humans don't contain that. So if someone tries to sell you something with a human pheromone in it in order to engage you in either intimate acts or anything like that, it doesn't work. That's not how it works. That's not how any of this works. Moving on to your sense of taste, this is also going to be a chemical sense. So keep that in mind, that your sense of smell and your sense of taste are both chemical based. These are going to be called gustatory receptors. That means that it is your taste receptors. This is different than a taste bud, so keep that in mind. Tastants are going to be chemicals that can be dissolved by your saliva. If you try to eat something or taste something after you dry your tongue, so maybe with a towel you pat your tongue dry and you make sure there's no saliva, and you put something on your tongue, such as like maybe sugar or cinnamon or something like that, onto your dry tongue, and you just hold your tongue out there, you're not going to be able to taste it. On your tongue, and if you have a camera or a mirror or somebody sitting next to you during this lecture, ask them politely to see their tongue. If you take a look at the surface of the tongue, you see all those ridges. Those ridges are known as papillae. These papillae are going to make up the entire surface of your tongue. There are different types of papillae, foliate, valate, and fungiform. And these are going to be located in different parts of your tongue, as you can see on this graphic here. And they serve different purposes. Some of them are going to contain taste buds, some of them are not. Moving on to what taste buds are. On each papillae, you're going to have 100 to 200 taste buds per papillae. This is where your taste buds are. When you have just a single papillae, that is not a taste bud. So everything you've ever thought you knew about your tongue is potentially wrong, and I'm here to set you straight. Within your taste buds, you're going to have these little taste pores, which are then going to kind of house these taste cells. Those are your gustatory receptors within there. So you can see from this graphic, going from the surface of the tongue to a single papillae, to the taste buds on the papillae, to a single taste bud, you have this taste pore where you're going to have those tastants come in and then cling to your taste cells, where they will then be processed. What's interesting is these taste cells are going to be replaced every 10 days. This makes sense that it would occur quicker than, say, your cells that allow for your olfactory receptors, right? Those replace every 60 days. Makes sense that taste cells would replace every 10, because they are going to be in this sort of line of fire, if you will, where there is constant motion, as well as saliva, enzymes, depending on what you eat, maybe acid, maybe more basic foods, it just depends. So it makes sense that they replace that quickly. So things that you're able to taste is bitter, sour, salty, sweet, and umami. And chances are you have a pretty good understanding of what most of those terms mean, aside from umami. Umami is going to be a term that refers to a fat or savory or meaty taste. The majority of your taste buds allegedly are at the margins of your tongue, although this little graphic over here isn't necessarily accurate. A lot of the science is sort of out when it comes to different regions of the tongue being specified to pick up those particular tastants. So bitter might be at the back of the tongue, umami might be down the middle, sweet is at the tip. It just isn't necessarily very clear whether or not this is all true, because there are taste buds that pick up these various tastants all around the tongue. It isn't just specified to one area. In order to taste better, you have a tendency to move food or drink around your mouth, so you can kind of cover as much area. When I eat my favorite snack, I have a tendency to suck on it for quite a while and move it around my mouth in order to get as much taste out of it as possible. And then you do the opposite when you aren't enjoying something such as medicine. You're going to potentially hold your nose And the reason you do that is because your sense of smell assists in your ability to taste things. Very heavily, in fact. Which this actually talks about it here in the middle of the page. So flavor versus taste. Flavor is going to be a combination of your sense of smell and sense of taste. Taste is going to be exclusively that salty, sweet, bitter, sour, and umami. But flavor is going to be kind of everywhere in between and combinations of. You can do this by doing something called the jelly bean test. If you have jelly beans of some kind, find a cherry jelly bean and a strawberry jelly bean. You can tell the difference between a cherry and a strawberry jelly bean because they have a different flavor. If you hold your nose while you eat one and then keep holding your nose while you eat the other, you're not going to be able to tell the difference. The flavor is going to be muted because you are not using your nose. The taste is going to be the same. It is going to be sweet. You will be able to tell that what you are eating is sweet, but you will not be able to tell the difference between the strawberry and the cherry jelly bean unless you unhold your nose. As I stated before, you need saliva in order to taste, so if you dried your tongue, you wouldn't be able to taste anything. Cravings like sugar, salt, and fat usually occur because your body is wanting more energy, but things like sugar are actually addicting, so when you think, I need something sweet, chances are it's just your addiction kind of saying, hey, I'm still here, feed my addiction kind of thing. And then things that kind of go into whether or not you like something or not, which can be genetic actually, but it is touch, texture, and temperature. So that is kind of, I can't stand things that are supposed to be warm to be cold. So things like soup or chili, I can't eat it cold, it has to be hot, otherwise it tastes wrong. I don't know if any of you can relate, but that's kind of what that is. Moving on to hearing. I want to start with some of the structures, and quite of this is going to go hand in hand with your lab, so maybe take note. But here I do want you to know kind of their function as well as what they are. The auditory receptors are going to be those sound receptors. Those are going to be within the inner ear, and we'll talk about where they are. The pinna is going to be this entire outside of the ear, and there are a few things that you can kind of go over, such as the helix, the concha, tragus, and the earlobe or lobule. But you don't need to know those, just know that the whole thing is called the pinna. This is the outside of the ear. You have the auditory acoustic meatus or auditory canal, and it's going to be this structure here. This is the part of your ear that you stick a Q-tip into. Of course, you don't want to go too far because then you will start to enter the middle ear and you could potentially hit the tympanic membrane or the eardrum. The tympanic membrane is going to be just this thin little membrane, kind of looks a little bit like a fan, which is connected to 3 of the smallest bones in your body. These are the auditory ossicles. The auditory ossicles are going to include, in this order, malleus, incus, and stapes. They are incredibly small, kind of cute for bones, but these are the smallest bones in the body. The stapes is going to connect to a part of your inner ear, but before we get there, I want to talk about this tube right here. This is called the eustachian tube. It is going to connect your middle ear to your throat. The purpose of this is to drain anything, whether it's wax or water or anything that gets into your ear, as well as to balance or equalize air pressure from outside your body to your inner ears. So you can do this or kind of visualize doing this when you yawn. You can hear like a pop sometimes. That's that pressure equalizing, as well as being on an airplane, chewing on gum or yawning a lot. And of course, as I say yawning, I feel that urge. Sorry. But as you're on an airplane, you feel that pressure changing as you are increasing in altitude. And what that means is that that pressure on the inside of your ear is wanting to kind of accommodate to the pressure that's outside of your body, right? So you can do that by yawning or chewing on gum when you're on an airplane. So back to our stapes and where it connects to this inner ear structure. So you have the stapes here, and it's connecting to something called the oval window. The oval window is then connected to something called the vestibule. The vestibule is connected to this thing called the cochlea. It kind of looks like a snail shell, and it's going to be filled with fluid. Next to, or kind of associated with, the cochlea is going to be the round window. The round window you can't really see very well on this graphic, but it would be somewhere about here. The round window serves the purpose, and this is a very important thing, to dissipate vibrations from inside the cochlea. Inside the cochlea, you're going to have these various structures called hair cells. These are going to be your auditory receptors. So remember these guys up here. Now, real quick, in a different color here, I'm going to go over the kind of start to finish how sound moves through the ear. Make sure you know this, and I do have a slide that does sort of go over this a little bit, but I like to do this while we have a nice picture here. So sound, as you're hearing me talk, is going to go into your ear via a wave. So you've heard of sound waves before. This wave is going to travel through your external acoustic meatus, or that auditory canal, after being funneled in by the pinna. So the reason your ear looks the way it does is because it funnels sound into this canal here. This wave is going to hit the tympanic membrane and cause it to vibrate. This vibration is going to be carried through your auditory ossicles until it hits that oval window. That oval window is going to vibrate. Now remember, the structure back here is filled with fluid. That fluid from that vibration is going to turn sound back into a wave. The way the cochlea is that it goes up and around like this. From there, that sound is going to come back down and around this little snail shell until it hits the round window. This round window is going to act kind of like a little foot giving, kind of like when you receive a soccer ball. So you wouldn't stick your foot out in order to get the ball would just bounce off, right? So what happens is that round window is going to give a little bit in order to receive that wave. Once it does, it dissipates the sound, or dissipates the wave, if you will. So here, I just want to go over that again, just super duper quick. You can kind of pass on this spot if you want to. Sound is going to travel into the pinna, also known as the auricle, but we'll call it the pinna. It is funneled in here. This is still a wave moving down the auditory canal. We hit the tympanic membrane. This is going to cause a vibration that goes through these auditory ossicles. We hit this little window here, the oval window, and this is going to cause a wave to move up our fluid here. And then we are going to hit that round window as it comes back down the cochlea, still in that fluid as a wave, and then it dissipates there. Now there's a couple structures that I didn't discuss that are in the inner ear. These are those structures. These are known as the semicircular canals. So this term here. Each canal is going to project into one of the three dimensions of movement. This is going to also contain fluid, As that fluid moves, it is allowing you to perceive that your body position is moving. So if I were to sit back, close my eyes, and spin my chair, I know that I am turning right and left. If I was on a roller coaster and closed my eyes, these semicircular canals would let me know that I'm going up or down, right, left, or upside down. So that is the purpose of them. And what we call this is equilibrium and balance. So equilibrium is your sense of balance. Static equilibrium is going to be your sense position of your head relative to gravity, or to sense acceleration or deceleration of the body. An example of this would be when you're seated motionless in a vehicle that is increasing or decreasing speed. You can tell that you're speeding up or slowing down. You can also say that an example of this is going to be when you're in a pool or underwater and you do a cartwheel underwater, you can tell that you're upside down underwater. Dynamic equilibrium is going to be maintaining balance when the head or body itself is rotated or suddenly moved. So again, sitting in a chair and suddenly, spinning around. This term down here, vertigo, is going to be what we call motion sickness, or inappropriate sense of motion. A good example of this is reading in a car. Your semicircular canals are going to register that you are moving, but your visual stimulation is going to say that you're stationary. So these conflicting things are going to make you feel nauseous, potentially, could make you throw up. So, moving a little bit closer into the cochlea is what this structure is showing us. So, the cochlea, if we cut it in half, kind of looks like a beehive, with these structures kind of holes here, which is just a continuous canal going up and then coming back down. So this is just one small section. And what we have is the vestibular membrane, the tectoral membrane, and the basilar membrane. But we do have these canals here that I do want you to know, the scala vestibuli, the cochlear duct, and the scala tympani. Now what's important about these is that they have different types of fluid in them. In the scala vestibuli and the scala tympani, you're going to have perilymph. In this cochlear duct, which is just this smaller portion here, is going to be endolymph. Make sure you know that. This is the fluid that is going to have that sound wave moving through it, and as it moves through, it is going to affect this organ right here. So this is the organ of Corti. As I said, this is kind of that view that we're looking at here, and you can see it a little bit better. You'd have that little organ of Corti right there. So the organ of Corti is going to contain stereocilia. These are going to be small little sound cells that are going to be able to pick up those waves and those vibrations kind of moving through. What's overlying the organ of Corti is the tectoral membrane, and embedded is the basilar membrane. So we have the tectoral membrane, and the basilar membrane is down here. Now these are the important little guys right here, these little hair cells. These hair cells, when waves are moving through this fluid, it's going to cause the tectoral membrane to bend, as well as the basilar membrane to bend, which is going to cause kind of a difference in these little hair cells. These little hair cells are going to be attached to nerve fibers, and that information is going to be passed on to the cochlear nerve, which is then going to be passed into your brain, and you perceive it as sound. That's all sound is just the movement and bending of these waves of fluid and these membranes, and specifically these little hair cells. That's all it is. Crazy. So this is that flow of sound through the ear. If you prefer things to be more written out, this is that. I do want to make sure that you guys kind of understand where hearing damage can occur. Most of these examples, you're not going to have too much damage, but one of the things I want to make sure that I discuss with you is this one here. So it says heavy metal rock concert. This is any concert, really. It could be Ed Sheeran. I wouldn't necessarily classify him as rock, but I do quite enjoy him. Regardless, that's not a rock concert necessarily. It is loud music or something very, very loud. This will cause, potentially, if you know what I'm talking about, if you've ever been to a concert, ringing in your ears after the concert. This ringing can last for a few hours, if not maybe a day or two. If you have ringing in your ears, you have caused irrevocable hearing damage. Those little hair cells that we were just talking about, those are going to be bent past the point of being able to repair themselves. Once it's gone, it's gone. So if you were to go to a concert or a shooting range or, I don't know, be listening to music, something like that, or be around, I don't know, a jet plane or a rocket launching, I don't know how many times you get the opportunity to do that, make sure you wear hearing protection. When you go to a concert, you may not feel very cool doing it, but you're going to feel pretty cool later in life when you can still hear and people around you can't. So just my little 2 cents. Wearing some hearing protection is important. A few things I want to go over as far as more about hearing is height and amplitude of a sound wave is going to determine its loudness or volume. The number of sound waves that occur during a specific time unit or frequency is going to be known as pitch. Cochlear implants are going to be those structures that sit on the outside of someone's ear, somewhere around here. What's interesting about them is they are going to correct some forms of nerve deafness. So this is specifically having to do with the cochlear nerve and not necessarily those little hair cells. Such as when you have hearing damage from a concert. There are different types of deafness, mild, moderate, severe, and profound, and the associating kind of definitions of those go hand in hand. Profound deafness is going to be absolutely nothing. Mild deafness is going to be just a very slight form of deafness. And then I do want to make sure you know this term tinnitus, not tetanus. Tetanus is going to be a bacteria found in rust. Tinnitus is going to be a ringing, buzzing, or swishing sound from no external source. This is going to come from within the cochlea. And that is basically forever. Once tinnitus starts, it is kind of damage to the ear. There's no way to fix it. And I've heard that it sounds like constant crickets in your ear. It's not pleasant. A few things about wax. It is ceruminous. It's going to come from the ceruminous gland. It produces cerumen or earwax. The purpose of earwax is going to be to protect the skin of the ear canal and assist in cleaning, lubrication, as well as provide protection against bacteria, fungi, insects, and water. I always think of a little girl that I babysat one evening. She was really sweet. She was like 7 or 8 years old. And in the middle of the night, as I was reading a book, she started screaming. I came upstairs and she was holding her ear. She couldn't tell me what was wrong. I called her parents as I took her to the hospital because I didn't know what to do. She just started screaming. What they found was an ant had crawled into her ear and got stuck in the earwax, and it was kind of moving and biting her eardrum. So kind of the purpose of earwax, and also sort of like a caveat, things get stuck in it, and sometimes you can have an overproduction of earwax, which can cause really large clumps of earwax and dirt to build up. One of the things I kind of want to point out, there's no real science to ear candling, which is what this lovely lady is doing here in this picture. It's supposed to pull your earwax and various things out of your ear, but there's not a whole lot of evidence to support that it actually does that. Last but not least, we have our sense of vision. let's go over a few of the anatomical and sort of the physiology of those anatomical structures. First, we have the sclera. This is going to be the white of the eye. So anywhere where you see white, that is that sclera. You have the cornea, which is going to be this projected portion at the front of the eye. So if you look at an eye from an angle from the side, you're going to see this kind of sticking out. The conjunctiva is going to be a transparent mucous membrane that's going to cover all of the eye and eyelids minus the cornea. So anywhere where you can see blood vessels, that is conjunctiva. The sclera is not vascular. The conjunctiva is. So if you've heard of the term conjunctivitis, that is the conjunctiva becoming inflamed. So this can cause like pink eye, or maybe if someone hits you in the eye, it is the blood vessels in the conjunctiva that are being harmed. Next we have the iris. This is the colored part of the eye. Whether your eyes are blue, green, gray, brown, or violet, that is the iris. The iris is going to surround the pupil. The pupil is sort of not necessarily a structure, but a lack of a structure. It is just a hole. It is going to go straight through to the back of the eye, but not before it passes through the lens. The lens is going to serve the purpose of focus and your ability to see distance. What controls the lens is the ciliary bodies. The ciliary bodies are going to be muscles that change the shape of the lens. So kind of pulling it tight or letting it relax. And that is going to control whether or not you're focusing on something far away or close up. Within the eye, you have a few layers. So the outer layer is the sclera. Remember, that's the white of the eye. From there, you have the choroid. The choroid is very important because it makes the inside of the eye look like a dark room. It's very, very dark. It is a dark lining. From there, you have the retina. The retina is going to contain your rods and cones. Your rods and cones are going to be your photoreceptors. We'll talk about that in just a minute. And then you do have the optic disc, which is going to be right here. Your optic disc is also known as your blind spot. This is where the nerves and blood vessels are going to come into the eye. I do want you to know kind of the process of how tears enter the eye, and I never want to hear you say, oh, my tear ducts are overactive. Your tear ducts don't produce tears. That's the lacrimal gland, the structure here that looks like kind of a chewed up piece of bubble gum sitting above the corner of the eye. The lacrimal gland is going to produce tears that is going to come through the lacrimal ducts, spread across the eye at a diagonal, and then filter in to the lacrimal canaliculi. The lacrimal sac, also known as the nasolacrimal duct down here, is going to be what kind of filters tears that are in excess into your nose, and that is why your nose runs when you cry. It is also going to allow for you to swallow your tears as well, so everything drains into your nose or your throat. There are two cavities for the eye. So if this is our eye, and we have our lens right here, and our iris, and our pupil is going through there, and I know that's a horrible drawing, but deal with it. Your anterior cavity is going to be from the cornea to the lens. Your posterior cavity is going to be this big portion back here. And the big thing that is going to be the difference between them is the fluid that they contain. Aqueous humor or vitreous humor. Vitreous humor down here. Your vitreous humor is going to be very jelly-like. The aqueous humor is very watery. And you can kind of ignore these two chambers. I don't really care about those. This vitreous humor is going to be jelly-like, and it allows for the posterior cavity to be inflated. If you did not have this vitreous humor, your eye would not be inflated, and it would kind of shrivel up into your orbit, and that's not a good thing. So here's this lovelier picture than mine. You have the posterior cavity and the anterior cavity. So make sure you're able to label those. A few organizations of eye structures and kind of how they work, very briefly. You have the cornea, again it is avascular, and the pupil. This is more important to me than that little bit about the cornea. The pupil is going to change shape, which allows for more or less light to come into the posterior chamber of the eye, or posterior cavity of the eye. So a dilation of your pupil is going to be that kind of blowing out of the pupil, becoming really, really big. Pupillary constriction is going to make your pupil very small. So you would have less light in order to make your pupil big, and more light to make your pupil small. The more light that goes into your eye, the more that light bounces around in the back of your eye. So your eyes function kind of like a shutter on a camera. You're trying to let more light in or let less light in. So when you walk outside from a dark room, or maybe like a movie theater, you walk outside and the sun's really bright. Your eyes are going to kind of like hurt a little bit because you're going from this dilated state where it was dark to this more constricted state because all that light is suddenly just pouring into your eyeball. Meanwhile, if you are sleeping and you wake up, you need a drink of water, maybe use the restroom, your eyes are going to look more like this because you are going to try to let in as much light as possible. So as I said, your rods and cones, part of the retina, are going to be photoreceptors. Your photoreceptors detect light. Your rods are going to be sensitive to light. Cones are sensitive to color. So think CC, cones, color. There are a couple portions on your eye that I want you to know. The macula luteae, but you can just call it the macula for now, is the region that is going to contain no rods. Within the macula, you have the fovea centralis, or just call it the fovea, is fine. It has the highest concentration of cones, so your highest area of seeing color. The optic disc is going to have no photoreceptors, that is why it's your blind spot. When you go to the optometrist, they are going to take a picture of something kind of like this. This is showing your macula luteae with your fovea centralis here in the middle. One of the things that your doctors are looking at is to make sure that your optic disc is looking healthy, as well as all of the blood vessels coming out of it. Now you can kind of flex on your optometrist next time and say, oh, how's my fovea centralis looking? Or how's my optic disc looking? Sound very, very smart to your optometrist. So how images are formed from seeing things is that light is going to form a tiny inverted image on your retina, and then the lens is going to focus on that image. One of the things I want to make sure you know is that the image that you're seeing is actually being inverted. So if it was a fence, you would be seeing it kind of backwards, and this post up here, you would be seeing it upside down. In order to kind of flip these images back around, the way your optic nerve works is from each eye, you have this nerve, and it actually crosses. So you're actually flipping that image around in order to go process it in the brain, in the very back of the brain, in fact. And then you also have this crossing here, so your left eye is actually being processed on the right, and your right eye is actually being processed on your left. Kind of cool. Brains. Weird. Stereoscopic vision is your ability to see depth perception. What I want you to know is that it requires two eyes with overlapping visual fields. If you do not have that, you are not going to be able to see very well. Your depth perception will not exist, and this can kind of play into whether or not you are allowed to have a driver's license or operate certain machinery. The terms hyperopia and myopia are going to refer to various stigmatisms within various individuals' eyes. Hyperopia is going to be farsightedness. This means that your eyeball is too short. Myopia is nearsightedness. That means the eyeball is too long. And then this also states how to correct. So concave lenses versus convex lenses. So if you wear glasses or contact lenses, it's kind of cool to take a look at whether or not you have concave or convex lenses in order to correct for that stigmatism. Moving on to the color of the iris. First and foremost, if you have blue eyes, that means you are a mutation. Isn't that neat? Blue eyes are something from your ancestors. There was a mutation that got passed down as a recessive trait. Originally, as humans developed, dark irises is what was the popular thing to have. It actually allows for less light to come into the eye. Blue eyes or light-colored eyes is going to allow for more light to enter the eye. This is why blue eyes can have a tendency to experience more problems, such as blindness, specifically when it comes to albinism. Blue eyes is what is expressed in that condition, and people with albinism actually go blind very frequently, unfortunately. But it is a genetic mutation, these blue eyes, or any color that's not necessarily brown. So it's just kind of interesting. And it all has to do with the amounts of melanin that you have. So remember, more melanin is going to be darker, less melanin is lighter. Last but not least, let's talk about colorblindness very briefly. This is a recessive trait that can run in families. It is more likely to be passed on in males because it is a sex-linked trait, meaning that from the mother and from your father, from the father, boys are going to get a Y chromosome, and from mom, you can get one of the X's. If one of the X's has color blindness, which I'll represent by this lowercase b here, and you get this offspring, and unfortunately when it comes to Y chromosomes, there's nothing all that interesting. You're going to get either this dominant trait or a recessive trait. So from mom, you can get this recessive trait here, and then nothing from dad, it doesn't matter. But a child born, a boy born with this recessive trait from mom, is going to immediately have colorblindness. So if a girl gets, maybe a lowercase b, so we've got a colorblind dad and a not colorblind mom, but she is a carrier. So we have a daughter who has capital B from mom, so she has that dominant trait, she's not colorblind, and we get that lowercase b from dad, she is going to be a carrier. But she won't express it, but her sons could. So this could be a potential son of hers. But what's interesting is that 8% of European men are colorblind. 75% of this group are red-green blind, which is what this picture is showing over here, what that looks like. This means that they have non-functioning red cones. This can be a result of non-functional genes that get passed on, which I said, as before, is recessive. Do keep in mind, you don't need to know this, just know that it is a sex-linked recessive trait. And I believe that covers special senses.